So it's a real pleasure for me to host Laura Buttitta. This was actually originally scheduled. I think we've been rescheduling this for a couple of years due to COVID and everything like that. So it's really wonderful to have it here in person and have her here meeting with people. So Laura started out actually got her PhD as part of Johns Hopkins and was at the Carnegie and actually worked on mice. But we will ignore that because she wised up and she moved to the Fred Hutch Cancer Center to work with Bruce Edgar in fruit flies and establish her real scientific question that she's been pursuing ever since, which is this idea of G0, both quiescence and postmitotic exit. And so this is a really, I would argue, important, but maybe underappreciated, at least to those of us who aren't cell cycle aficionados, part of the cell cycle. So there she was awarded K99R00 and was recruited to the University of Michigan in 2011. She was promoted to associate professor in 2016, and I know that her department has voted her up for full professor. So while it's not official, I think that's worthy of mentioning. And she's won a number of awards, like an ACS Scholar Award, a Prostate Cancer Foundation Award, and also really impressively from her CV, she clearly has a commitment to mentorship and service. She's mentored over 30 undergraduates and had a number of postdoc and PhD students who have all gone on to diverse and exciting careers. And she also has a huge amount of service that I won't list because we'd be here forever. But more on a personal note, I first met Laura when we were both starting our labs in 2012 at the Drosophila Research Conference. And Laura is one of those people who is both a scientific colleague, but also a friend. And so I like to highlight this because I think sometimes a really underappreciated part of the amazing part of this job is that we meet people from all over the country who are smart, passionate, do fun science, and are really solidly good people. And I really count as a friend, someone that I will text when I'm going through a tough time, not just about science, about life. And the sort of friendship was solidified because I think in the first few years of our career, like every year or more than once a year, we ended up at the same conference. And so at some point our scientific, it was either we were going to become mortal enemies or best friends. And so we went for the friend option. And if you know Laura, I can't imagine her having a mortal enemy. And so with that, I will let her tell you about her fun and exciting science.
Thank you so much. What a fun day for me to be here. This is my first in-person visit and seminar since COVID. So thank you so much, everyone, for being here. And I've had a really great day talking with everyone here. I was here in 2014. Some of you may remember. And I'm going to tell you a very different story since our work has progressed since then. But it's been fun to get a chance to visit again with people that I met during that first visit, but also a few people that have known me since the 90s. There's a couple of people here at Wisconsin now who were at the Carnegie when I was a young grad student. And so what a fun extra pleasure to get to visit with you guys today. So my lab is at University of Michigan. We're in the College of Arts and Sciences called LSA, Literature, Sciences, and Arts. And so my lab, we have a lot of undergrads because in part my department is part of the group that administers the undergrad biology major. And then we also have our own graduate student program and postdoc recruitment programs. So the work in my lab has evolved over the last few years as I've recruited new people into the lab. And so I'm going to tell you a story that's going to have cameos from a lot of people in my lab. A couple have graduated and then a few are new. So I did switch to the Drosophila system in part because of the beauty of the system and also the tools. And so the question that my lab is interested in addressing is trying to decipher this mysterious state of the cell cycle called G0. And so I've drawn the cell cycle this way here because I wanted to illustrate with these dotted lines the fact that G0 actually is a phrase that gets used to encompass many different states of cell cycle arrest. So for example, you can imagine a quiescent cell that may leave the cell cycle and enter into a non-cycling state of G0 for only a temporary amount of time, but then perhaps you add serum or a growth factor and it readily re-enters the cell cycle. This would be in contrast to cells that may be more refractory to cell cycle reentry. Perhaps in this case, you might need some kind of damage or some sort of more acute stimulus to get those cells back into the cell cycle. And then we can also think of cells that under normal conditions would be permanently postmitotic, or so we think. And, examples of these might be multinucleated muscle cells, perhaps cardiac, terminally differentiated muscle, or neurons, like postmitotic differentiated neurons. These cells enter into a G0 state, they exit from the cell cycle, and in most cases, although we will talk about exceptions, they're not going to re-enter the cell cycle. And so this would be a state of a permanent G0. And so this is sort of an issue because this means that this term G0, if we want to try to understand what it is, it's encompassing many different states. And if you ask somebody in the cell cycle field what it is that regulates exit from the cell cycle in G0, whether they're working on reversible quiescence, senescence, or terminally differentiated cells, they will tell you the same thing. Cyclin CDK inhibitors, RB-E2F complexes, which are repressive gene expression complexes that turn off cell cycle genes. And so we know that those can't be the only mechanisms promoting this G0 or influencing the flexibility of G0, because then G0 wouldn't have all these states with very distinct capacity for cell cycle reentry. So we think that there must be like more to this code of, the state that's promoting exit from the cell cycle and making it more or less refractory for cell cycle reentry. So that's what these dotted lines are intended to indicate. The blue things are indicating sort of projects that dance around this state of G0 in my lab. And unfortunately, I won't have time to talk to you about all the stuff we're working on today, but a couple of things that we're working on are trying to understand how DNA packaging and chromatin changes when cells exit from the cell cycle and enter into G0. And as cells are in G0 longer, their quiescence deepens. This has been seen by a number of labs working both in cell culture, but also in vivo models of G0. And so trying to understand that is one aspect of the lab. We're also interested in developmental signals that might regulate either exit from the cell cycle or re-entry from a G0 state. And then I'm going to tell you a fair bit of work that we've been doing on trying to understand how aging and DNA damage might impinge on neurons to get them to re-enter the cell cycle from G0. So this would be a cell cycle re-entry story. So these are some of the players for my lab, and Liz and Ellie are working on the chromatin packaging aspect of trying to understand what happens to chromatin as cells enter into deeper and deeper states of G0. And so we sort of can divide up the lab into projects focused on entry into G0. And then we sort of have this bigger group of people, it's growing in my lab actually, that are looking at exit from G0. And so Jamie and Allison and Navia, I'm going to tell you a bit about their work today on looking at the Drosophila prostate and a situation there where the cells are quiescent, but then can re-enter the cell cycle. And then Shama Nandakumar was a graduate student in my lab, but she actually graduated in August 2020 and has moved on to a postdoc position. But I'm going to tell you a story that she started and really developed in my lab and is now being taken over by our current postdoc, Dina Damschroeder, who just joined my lab a year ago. And so just to illustrate to you some of the things that we're interested in is we want to understand how the signals of damage or developmental signals might impinge on the cell cycle. And we ultimately think that some of these signaling events are happening through changes at the level of the chromatin, where you actually get either transcriptional repressors binding, or you have actually remodeling of chromatin accessibility itself, such that enhancers that are temporally dynamic at cell cycle genes, in this case, the Drosophila gene string, close when the cells enter G0, and then we think that they're reopening when the cells re-enter the cell cycle. And so this is kind of an example of how we're using molecular tools to bridge the gap in these two projects to understand how the chromatin organization changes in G0. So the first story I'm going to tell you about is our story where we're looking at the fruit fly brain. And I should give a full confession that I have absolutely zero neurobiology training. So the way that we ended up looking at the fruit fly brain was that we were looking for a tissue that was homogenously in G0, like there would be no cycling cells. And we wanted to use this as sort of like a tissue type where we could have like cell type specific drivers and we could re-express cell cycle factors and push them back into the cell cycle. That was the original idea. And so we were doing some experiments where we were dissecting the fly brain. And we first just wanted to establish that, you know, that there was no cell cycle and no proliferation in the adult fly brain. And at the time we were using a flow cytometry assay to do this. So the way that this works is we dissect the fly brain. Of course, our forceps are quite a bit smaller than this one. We dissociate and stain the cells with a DNA dye. You can use DAPI or you can use dicycle violet. We've done it both ways and you get very similar results. And then you run the cells on a flow cytometer and you can look at the ploidy of your dissociated cells by plotting the DNA content versus number of cells. And so if we plot it as a density plot where each dot represents a cell, you see that all the cells kind of pile up along this. This would be so-called forward scatter or a proxy for cell size. And then the x-axis here is going to be DNA content. And you can see that when you look at these dissociated cells, almost all of the cells have a very similar looking DNA content. They all pile up in a line. And if you plot this as a histogram, you can see this nice sharp peak of cells that have a 2N or a diploid DNA content in the brain, which is exactly what you would expect for the fly brain, which has very few stem cells. The adult fly brain has very, very few stem cells. And the levels of cell proliferation in the adult brain under normal circumstances are like basically undetectable. So, okay, we thought that, so we were surprised when by accident, we took some aged fly brains. And so these are brains from flies that are 56 days old, which if you haven't worked with flies before, this is a pretty old fly. They start to die around 70 to 80 days. So it was an old fly. And we were surprised to see that when we looked at the DNA content in these aged brains, we saw other populations of different ploidies pop up. And you can see this in the dot plot as these lines that kind of are falling in multiplications of the genome size. You can see them as additional peaks here in the histogram. And so the first thing we thought was like, okay, this is like some sort of artifact of our dissociation. Maybe these are cell clumps or doublets, things like that. We spent our first actually couple of years on this project trying to ensure that this wasn't just some sort of artifact of our dissociation process or you know, something that we were doing strange in the flow cytometer. And so we went through and we strained cells, we did doublet gating, we made sure that we were fully dissociating the tissue. And every time I started to walk away from this project thinking it was an artifact, I would get some result that would bring me back to looking at it again. So long story short, we basically kept reproducibly finding that polyploid cells were accumulating in the aging brains, which was a surprise to us because the neurons and glia in the adult fly brain should be completely quiescent and exit from the cell cycle. So in work that Shama Nandakumar did in her grad school days, this is, she was looking at how the ploidy, the accumulation of the polyploid cells happens in the brain with age. And I just want to back up by saying this is, of course, a small percentage of cells. So most of the cells in the adult fly brain do not become polyploid. It's a small percentage or fraction that do. So she measured this over time and she looked in Canton S and White 1118. And so these are two Drosophila fly stocks that the community commonly uses as sort of wild type controls. The thing is that Canton S flies have a wild type eye color, nice bright red eye, whereas the White 1118 is this mutant that lacks eye pigmentation. And so we just wanted to make sure it wasn't like an artifact of looking at flies that don't have eye pigmentation. And then we also separated brains into male versus female because we weren't sure if there was some sexual dimorphism in the phenotype. And so these are the plots that she shows. And I should tell you that these were run on individual brains. So part of what these error bars are capturing is the individual variability from brain to brain. In later experiments, I'm going to show you examples where we pooled brains for the flow cytometry. We actually found that whether we pooled brains or whether we ran them individually didn't really have a huge impact on the level of variability that we saw. So what you can see looking at this percent, and at this point we were calling it hyperploid because all we know from the flow cytometry is that there's additional DNA content. We don't know if it's genome-wide duplication. We don't know if it's specific chromosomes, what it is. So at this point we were just calling this hyperploidy to indicate that it was more than a diploid DNA content. So what Shama found was that there's an increase in the percentage of the hyperploid cells up into about 21 days. And then as we look at later time points past 21s, it kind of levels off and we don't see a continued increase. And so one of the issues that often comes up here is like, you know, a 21-day-old fly, is that really old? And so by 21 days in the fly lifespan, you're already starting to see some fertility decline. You're actually already starting to see fragmentation of the circadian rhythm and some other hallmarks of aging. So even though the fly can live to like 70 days or so, even at 21 days, you are starting to see the beginnings of some hallmarks of aging. So I do agree that that's a interesting discussion that we could have, would be like if this is really an aging phenotype or a developmental phenotype. And so just to give you a sense of where this is in the lifespan, so these were Shama's lifespan curves for these flies. And so, you know, we're seeing, at least in our hands, we're seeing decline of lifespan between, you know, 56 to 70 days. And this window in which I was just showing you the polyploidy is really here. So this is before we're starting to see a lot of flies dropping dead. So the problem with flow cytometry is the first thing you do is you dissociate the tissue. So we had no regional or region specific or cell type specific information. So we wanted to try to see if we could localize where the polyploid cells might be coming from. So Shama did a dissection of the brain, and she dissected the optic lobes, the central brain, or the ventral nerve cord, and she separated the samples out, and she looked at them at 7 days, 14 days, and 21 days. And what we found was that the majority of the hyperploid cells are coming from the optic lobes. And this was the case whether we looked at Canton S or white 1118. So it wasn't dependent upon whether they had the mutation in the eye pigment color. And for the sake of time, I'm not going to show you the data, but I'll just let you know we had an honors student who worked with Shama on a project where he looked at different mutants that failed to form a retina. So we looked at glass mutants and a number of other Drosophila mutants where they basically have no eye, but they still have an optic lobe. And we actually saw that you don't even need to have a retina form in order for this polyploidy or hyperploidy to accumulate in the optic lobe. So we think it's not dependent upon the actual function of the visual system. We've never actually tested something such as like hyperactivation of light sensing or things like that. So there's more to explore here, but we know that you don't need to form a fully functioning retina to get this phenotype. So that told us that they were in the optic lobe, but we wanted to actually see these polyploid cells. And that was sort of one of the things that, like, for us was tripping us up for a long time, because, you know, seeing hyperploidy in facts is one thing, but actually physically getting to see where the polyploid cells are is something else. And so one of the challenges for us was that when we tried looking at different cell type specific drivers, so we tried drivers for glia, we tried drivers for neurons, we tried drivers for subtypes of neurons, such as glutamatergic neurons, et cetera, other classes of neurons. And what we found was that we would see some fraction of polyploid cells or hyperploid cells using all of those drivers. So we think what's happening is that there's a subset of glia that become hyperploid, there's a subset of neurons that become hyperploid, and those can fall into different classes. So we started to wonder if maybe, like, if we could visualize these cells and really, you know, look at their nuclei and confirm, like, were they binucleate, were they multinucleate, what was going on here? And so we didn't have a way to identify the polyploid cells. If you ever tried to do nuclear staining on the Drosophila brain, you know that the nuclei in the brain are all packed really closely and tightly together on the surface of the tissue. So it's really hard without some landmarks, some staining landmarks to distinguish, like, you know, ploidy of individual nuclei just by looking at a DAPI stain or a nuclear laminin stain. So we use this tool called CoinFlip, developed by Ishwar Hariharan's lab, as a way to try to mark the polyploid cells. So let me explain to you how this works. So CoinFlip is this binary flip out cassette system where you have two flanking but unique FRT sites. So the gray FRT or flip base recombination target sites, the gray ones can only recombine with gray, and the yellow ones can only recombine with yellow. And so if you get a recombination between the gray, you flip out this stop codon, and then you express the LexGAD, and then there's a stop at the end of this LexGAD. If you flip out the yellow, you flip out the stop and the LexGAD, such that now you can only express the GAL4. And so you can take this construct and cross it to flies that carry a Lex AO responsive GFP or a UAS responsive RFP, and you can get this color choice where cells will either flip red or green. And this was what the Harry Heron labs used in a paper, coin flip. And they called it coin flip because the idea was like it was a 50-50 chance that you would flip either red or green in this system. Now, the key thing here is that you have the construct heterozygous. So if you do the cross so that this construct is heterozygous, you only have one copy of the construct in the diploid genome. But if you become polyploid, you can end up with 2 copies of the construct, and a third of the time you can flip 1 copy red and 1 copy green. And so the presence of yellow cells can indicate for you that there are polyploid cells with multiple copies of the genome. And so we looked at this construct on the 2nd and on the 3rd. And so this is where we started to, and I'll show you what we saw. So we saw that we could identify RFP, GFP double positive cells. They have kind of funky lamin staining. Like we often would see that they would have strange shapes when we looked at the nuclear lamin staining. Here's the nucleus for one of these, and you can see we saw a lot of weird things. So this one has like this super huge heterochromatin looking ball of heterochromatin. And then this one here, if I take away the outline, it's easier to see, but you can see it looks like there's like sort of like this, I don't know, almost spooling of DNA, like this big blob is all together within one nuclear lamin region. So some like decondensed regions, some expanded hypercondensed regions. We saw some funky things like that. More importantly, when we did coin flip and we actually did DNA quantification of the DAPI, so now we're using the coin flip as a landmark to tell us which cells actually have multiple copies of the genome. When we do that DNA quantification, we see that we get ploidies from our DNA quantification from images that look hyperploid. So if we look at the coin flip yellow cells, they all fall between like a 3N to higher ploidies. But if we just take measurements of all cells, like we just say take 100 nuclei, we don't pay attention to whether they were coin flip positive or not. This is the profile we see. So most of the cells are diploid. So coin flip is helping us to pick out a subset of these polyploid cells. And I just want to say it's a subset because, of course, if I go back here, you could also imagine that you could flip red, red, or green, green. And we wouldn't, I mean, we could sort of thought sometimes we could see brighter RFP or brighter GFP, but we wanted to be rigorous. So we didn't take those into account, the cells that could potentially be red, red, or green, green. So right now we're just looking at the yellow cells, but that's one caveat of this system. So this convinced me finally that we actually had polyploid cells in the brain. And so now that we had coin flip kind of guiding our eye, we sort of became aware of patterns of nuclear lamin staining that we could see, that we could like reproducibly see when we stained brains. And so I'm just showing you a smattering. This is from a really great grad student by the, so she finished her master's at MCB, but she's applying to PhD programs and she's really great. So her name is Emily Rosich. So she was in my lab for 4 years. She's absolutely awesome. She did a lot of staining on brains, and we sort of can start to pick out. So here's an example of an LV negative cell that looks like large nucleus. Here are some examples of LV positive cells that have large nuclei. And here's another example here. So we're starting, and this is without the coin flip marker. So we don't have direct molecular evidence that these are polyploid cells, but we're thinking that these are polyploid cells based on how the polyploid cells that we identified with the coin flip method looked. So we're thinking we're gonna maybe be getting close to being able to identify these cells without using the coin flip marker. And so the next question was thinking about, you know, if we just take these ploidies without the coin flip marker and then we measure their ploidy, can we discern any sort of polyploidy with the DNA content measurements? And we can. And so what I'm showing you here is just where we measured LV positive neurons. So there are examples that are already known of glia becoming a polyploid in the Drosophila brain. What we identified was many, many more classes of glia exhibiting polyploidy than had previously been known. But what we were the most intrigued about were these polyploid neurons, because that just shouldn't happen, or at least that's what I thought based on, you know, my education. So we were really trying to drill down on these LV positive neurons and make sure that indeed we can see that these LV positive neurons that look polyploid when we measure the DNA content from the image analysis, they look like they're polyploid. And one thing I'd like to point out is that the cells where we see really high ploidies, like 8N and higher, those tend to be LV negative. We think that they're glia. The cells where we see mostly tetraploid and intermediate diploid tetraploid DNA contents, those tend to be neurons. So it's almost as if the neurons can do a little bit, but the glia can do a lot more of this abnormal cell cycle where they're becoming polyploid. Okay, so why would the cells do this? We didn't know, and we were hoping to find markers of polyploid cells. So we did an experiment that we didn't have the best rationale for, but we just said, okay, well, let's take the regions of the brain, let's do RNA-seq on a time course, and we did multiple time courses. We divided up the brain into optic lobe, central brain, ventral nerve cord. Oh my. Do I have until 4:30? Yeah. Okay, great. And we did a time course of the adult brain. And I will also tell you, for anyone who might be interested in this data set, it's like tons of information. We kind of don't know what to do with it because, as I said, we're not neurobiologists. But we also separated it out by male and female. So we also have sexual dimorphism, changes in gene expression with age in the various regions of the Drosophila brain. So reach out to me if anyone's interested in looking at this data set. The one thing that jumped out to us, though, was that when we looked at the optic lobe, when we looked at 3-week-old versus 2-day-old brain, so this is looking at the peak of the polyploidy versus the newly emerged flies that show almost entirely diploid DNA content, we got this upregulation of genes that are involved in DNA processes, cell cycle, DNA recombination, meiotic, double-strand breaks. So this big signature of DNA damage. We also, I mean, the strongest thing we got was chaperone-mediated protein folding. So we are seeing proteostasis defects coming up in a 21-day fly. Like a lot of these genes are known to be involved in like some of the proteostasis issues that arise with aging. And so that was our strongest signature, but very close behind that was like mitotic cell cycle, meiotic cell cycle, double-strand break things. So we think that what's happening is that the neurons in the brain are long-lived, and so they're accumulating DNA damage. And much of your DNA damage repair pathway is actually under the control of the cell cycle. So many of the genes that are involved in DNA damage repair are actually targets of a transcription factor called E2F, which is the main transcription factor involved in controlling cell cycle gene expression. So we were thinking that maybe what's happening is that neurons need a way to repair damage and accumulation of double-strand breaks, but they really only have non-homologous end joining at their disposal because they're not in the cell cycle, so they can't do other types of double-strand break repair. And so maybe they reactivate this pathway partially just to be able to cope with the accumulation of DNA damage. And so we thought that if that was the case, that if we induce DNA damage, we should see more polyploidy earlier. And so we took 2 ways to do that. Here, what we did was we UV treated the flies, and this was great because if any of you guys have an old stratolinker in the lab that you haven't used in 10 years, you can pop flies into that stratolinker and you can UV damage them really reproducibly. It was revitalizing a piece of old equipment we almost got rid of. And so if we UV damage the flies, even at 1 week, we see this jump in the percentage of the brain that becomes polyploid. We did another approach. This is where we actually fed the flies paraquat, and in this case, so there are some sexual dimorphic responses to oxidative stress. I'd be happy to talk to anyone wants to. So I'm just showing you the data from male flies for now, but you can see that there's this accumulation of this increased accumulation earlier at 1 week for the males treated with paraquat, whereas the control males just given the vehicle, you see the normal like sort of increase over 21 days and then it plateaus. So we think that accumulation of DNA damage, and this could either be by our UV treatment or indirect DNA damage induction through the oxidative stress, it can increase the polyploidy. But we consider another possibility, which was that maybe it's just that like if you have cells dying in the brain, the remaining cells have to compensate for their loss. And so maybe it's not really that we're inducing DNA damage, it's that we're killing cells. So we wanted to do another approach that would kind of kill cells in the brain. And so this was done by a really great honor student, Chelsea Yu, who graduated and is now in med school at Wayne State. And what Chelsea did together with Shama was, this is our control experiment. They expressed different UAS-inducible neurodegeneration proteins in flies. And so there's, for example, like human Parkinson's associated TBP43, tau, Huntington, transgenes that people have used in flies to cause neurodegeneration, mostly in the eye and in some cases in the brain. And so what they did was they expressed these in the brain, and they used a cell death assay that we use in the lab where we expose the dissociated cells to propydium iodide. The cells are alive, so if their membrane is intact, they don't incorporate the propydium iodide, but if they are dead or dying and have compromised membrane permeability, they incorporate the propydium iodide and they turn what we call PI positive, which means they're dying. And so we can measure that and quantify that in our flow cytometry assay at the same time that we quantify the polyploidy. And so long story short, what we found was several of these, maybe not this one so much, but several of these induce an increase in the cell death in the Drosophila brain when we express them, but we didn't see a consistent increase in the polyploidy. So that suggested to us that it's not just simply loss of cells that's inducing this. It might be something specific to either the oxidative stress or the DNA damage. But this is unpublished. This is like preliminary. For the recording, this is preliminary. So then we thought, well, why would the cells do this in the brain in response to DNA damage? And we were really inspired by work from Brian Calvi, who had previously shown that polyploid cells in Drosophila were actually refractory to apoptosis. And in fact, what he had seen was that cells that endocycle become polyploid, that they epigenetically silence some of the components of the cell death pathway. So we thought maybe the polyploidy could protect from cell death. So if we just do our normal cell death assay in whole brains, we see a very low level of dead or dying cells that are PI positive. And we do see that most of them are diploid. But these are like very small numbers. It was not convincing to us. So we wanted to look in a background where we had actually increased cell death. And so here what we did was we UV treated the flies. And I should point out that in these experiments, we're looking at pooled optic lobes only because we were trying to look at as many of the polyploid cells as we could. And so what we see when we increase the level of death by treating them with UV, we can pump up this level of cell death to like 20% of the cells, start incorporating propydium iodide. And we see that the majority of those cells are diploid, almost suggesting that the polyploid cells are protected from cell death. And so we're thinking that maybe this could be neuroprotective, which is unusual because a lot of the arguments about aneuploid and polyploid cells in the brain suggest that it's associated with neurodegeneration, but we're thinking perhaps it might serve a neuroprotective function initially. But again, this is preliminary. We're still trying to understand this phenomenon. I will say that this response to the UV damage, we kind of had to characterize it. So other work had looked at exposing flies to radiation to create double-strand breaks in the brain. And they had seen that there was no cell death in response to the radiation. And it was actually a really interesting paper because they had pointed out that they exposed the flies to as much irradiation as they do to like embryos or wing discs, and they could see abundant DNA damage, but they didn't see any evidence of cell death. Whereas we were exposing to UV, we see this peak of cell death 24 hours post-exposure, but then it returns to normal relatively quickly. So our UV treatment is causing an acute damage. It's causing a short-term wave of cell death, and then the brain returns to normal. And I will say the difference in our study was that they looked at 16 hours post IR exposure. And I think it could be that the timing is different. And then the other issue is that the reason why they looked at 16 hours post-exposure was because all of that response was based on work done in the wing disc and in the embryo. And we just think that the damage response in the brain has like different kinetics possibly than in those other tissues. So like we need to do more on this time course. We don't know if we're hitting the middle of the peak here at 24 of the cell death, or if we're just catching the tail end, or if we're catching the early beginning. So there's a lot more to fill in this question for this model of cell death. Okay, so maybe it's neuroprotective. That was a little bit what I was telling you here, but I'll tell you that we know that it's not always neuroprotective. So in another experiment where we drove cell cycle reentry in pupa, brains. We used an NSIB Gal4 driver to do this. So we're driving cell cycle reentry in neurons, if I remember correctly, on this experiment. And we can clearly see that we are driving like multiple ploidy populations. We backdated here the cells to be PI positive in pink, and you can see that when we drive polyploidy, like by forcing cell cycle reentry in postmitotic neurons in the pupa, that they actually die. And we did this with some specific neuronal classes like PDF neurons, and we can see that they like totally degenerate if we drive cell cycle reentry in the pupa. So why would that be, that driving cell cycle reentry in the pupa is detrimental, but this sort of natural physiological cell cycle reentry in the adult is perhaps protective? We really don't know. This is like, I'm sharing my confusion with you at this point, but this is kind of where we are with our understanding of this unusual and surprising phenomenon. So our overall thought about this is that, you know, as the animal ages and these neurons have to be long-lived, they accumulate DNA damage, the oxidative stress can feed into that, and this can lead to cell damage or loss. And then if the cells are dying, perhaps they non-autonomously signal to the neighboring survivors to increase their ploidy, which now will protect them. That's kind of the model that we have. But obviously we have a lot of detail and molecular detail that we have to fill in here. So this is really an emerging project. So that's an example where cells are postmitotic. They're supposed to be in a robust and permanent G0 state, but they seem to be re-entering the cell cycle and becoming polyploid. There's another example of this that we're studying in the lab, and this is the Drosophila accessory gland. And so for all of you guys working on fruit flies, your male fruit flies have prostates. It's called the accessory gland. And it performs the same function for the male fly that the prostate performs in mammals, which is that it secretes the enzymes and proteins and components for the seminal fluid for fertility and transfer to the female. And one of the cool things about Drosophila is that the seminal fluid proteins have been studied in some level of detail on an evolutionary scale, because Drosophila do sperm competition where the female can multiply mate. And then she stores sperm from multiple matings, and then the male sperm actually competes to like be the one to fertilize the egg. And so Drosophila have all this interesting evolutionary biology and genetics where the males keep trying to evolve proteins that help them to outcompete their competitor's sperm. So kind of cool side story there, but very little had been done to actually look at the cell biology of the accessory gland, like what the cells are like, and also very little had been done to understand their cell cycle regulation. And so we were kind of curious about this tissue, because hopefully one thing that's striking you about this confocal image of the tissue is that the cells are polyploid and binucleate. So it's a really funky cell cycle situation where there's two nuclei. Currently in the literature, people think that those two nuclei are 4N. We actually know that that's not the case. We're working on revising our current preprint, but we now know that because these cells undergo 2 rounds where they partially under-replicate their DNA, they're actually more like 6N. So very unusual DNA ploidy situation. And then the binucleate polyploid situation is kind of cool and funky from a cell cycle point of view as well. And so what you're looking at here are these secretory cells. They have like sort of a hexagonal shape, lining of lumen, where the apical surface, they secrete all their proteins for transfer to the female. And this is what the system looks like. So what we're looking at is the accessory gland here, these 2 lobes. They empty into this tissue called the ejaculatory duct. These are connected through a zone of tissue we call a transition zone, and then the testes are of course providing the sperm. And so in our manipulations, we don't manipulate the germline at all. We're just looking at the somatic portion of the male reproductive system. So flies are a great model for cancer. One of the things that I teach at University of Michigan is cancer biology. So one of the questions we had early on was, can we model any aspects of prostate cancer in the fruit fly prostate? And so we started out just by overexpressing MYC and activating the cell cycle with E2F. And what we found is shown here. So I'm showing you just to reorient you to what you're looking at. You're looking at something kind of, although maybe the lobes didn't fall quite with this upward location, but this is the duct here, the 2 lobes here. What you're looking at here, here's one lobe. Here's the ejaculatory duct. Here's the other lobe. And so the normal epithelium looks like this, where it's got this nice polarity. We're looking at a septate junction protein DLG in white, and then the nuclei are in magenta. and this is the lumen. When we overexpress MYC and E2F in a subset of cells that are labeled with GFP, we get these massive overgrowths where the nuclei are absolutely enormous and huge. The ploides are like incredible, and they invade the lumen or they start to extrude basally. And this is all in the absence of mitoses. We've stained tons of these with phosphohistone H3. We see no evidence of mitoses. So these extrusion events and these overgrowth and hyperplasia events are all happening in the absence of mitosis. So, okay, and not maybe a super great model for human prostate cancer where mitosis is part of the phenotype, but we can model things like the basal extrusion, the luminal invasion, stuff like that. But then we were thinking about this, that these nuclei are absolutely enormous. Their ploides get enormous. And we knew from other experiments in my lab where we had overexpressed MYC and E2F and many other cell types in the adult that normally terminally differentiated postmitotic cells are refractory to re-entering the cell cycle in response to just simply adding more E2F and MYC. So why are these oncogenes, E2F and MYC, driving cell cycle reentry and hypertrophy in this tissue? And so we started to think that these cells might remain poised to re-enter the cell cycle. And so that became a state of G0 we were interested in. The cells were not normally cycling, but they were poised to re-enter with a relative, what we consider in my lab to be a relatively mild stimulus. And so then we thought, well, what would happen if we damaged the gland? Maybe they have some capacity for regeneration, and that's why these cells remain poised, is because like when the male mates, maybe there's some level of damage or cell turnover or something. So we used some flies that was kindly shared with us by Don Fox's lab at Duke University. That they use a line called demise, which is a genetically inducible damage system where you can turn on cell death proteins, and in Drosophila it's called Reaper, like a caspase cascade. You turn it on with a heat shock, and then by shifting the flies to different temperatures, you can turn on or off the damage. And one of the important things about the damage is that it happens sporadically throughout the tissue. And so we used demise, and indeed we saw that they can recover after damage. And so if the undamaged tissue looks something like this, when we cause the damage, we see that these accessory glands are small and shrunken. We see tons of pycnotic nuclei. We see DCP1 positivity, like a cell death marker in the tissue. And then when we put them at a low temperature, we see that they can recover. We see a restoration of some of the gland size. The cells become huge in the recovery condition, and we see EDU incorporation. This is all without us doing any cell cycle manipulation. So the only thing we drove here was induction of apoptosis. And so this suggested to us that indeed the cells are poised to re-enter the cell cycle in response to damage, and that they're undergoing this process really nicely characterized by Wu Min Bing's lab called compensatory cellular hypertrophy, such that after you lose a fraction of cells, some of the cells will re-enter the cell cycle and become hypertrophic to restore from the damage. And this work was carried out by my grad student, Allison Box, who will probably be defending next year. So work for her for a postdoc position. Ooh, sorry. So what does this look like? So I'm just showing you a little bit here, but it becomes much clearer here. So first of all, the level of damage is variable depending upon how we do the temperature shift and the degree of heat shock. So if we have a situation like this, so again, I'm showing you the cell junction proteins just so you can see the size of the cells. So here's a condition, a control where we didn't do any damage. We can do damage and recovery, and we'll see, for example, sort of 3 different levels of recovery. Here we saw 25% of the cells showed increased ploidy based on our DNA ploidy measurements. We can get something intermediate with 32%. And then this one here was pretty severely damaged. The cells become absolutely huge, and 86% of those cells show an increased ploidy after the damage. And so we can also look at the cell number per frame to get a sense of the cell size. And so we're working on like quantifying these more carefully. And so the gland size after we induce the damage, so we see that almost all of the glands show reduced size during damage. We do have this subset up here where we see damage, but the gland size overall isn't necessarily smaller. And then during recovery, we see most of the glands recover their size. We do see, again, a small subset fail to recover fully. And so that's true. It's not like a 100% system, and we're still working on, precisely characterizing the timing of the heat shocks and the temperature conditions to get this like as clean as possible. But the big thing is the fertility. So if you want to know if it's actually regeneration, you need to demonstrate or have some assay for functional recovery. And so when we look at the fertility of our males, this is our 25-degree control. I don't know what's up with this male, but clearly we need a bigger sample size. The males under demise, where they're getting cell death, they have reduced fertility. Again, we do have some variability. When we put the flies back at 18, so the problem is Drosophila, and this is well known, have reduced fertility at 18 for a multitude of reasons, both on the male and female side. So you have to compare this plot to these guys here, because this is the rate of fertility at 18. We see that 3 out of 4 have recovered their fertility. So we think that this could be a new model for functional regeneration, and we're continuing to quantify this better, have more replicates, have a more precise damage paradigm, so that we could make this like a real model of postmitotic regeneration. One part of that is going to be to do a genetic screen. So I'm a Drosophila person, so the most powerful thing we could do in this case is probably a genetic screen. So we wanted to have a line where we could follow damage and recovery in the same fly. So one thing that might be bugging you about what I just showed you and kind of bugs me is that we can't like follow, well, these ones we can follow the same fly, but like this, in order to look at the size and the recovery, we had to dissect the fly, right? So we couldn't track the damage and recovery over time in that situation. So what we did was a cross where we put together the driver for the demise system and the heat shock inducible demise together with a UAS driven RFP, so that it lights up the accessory gland internally with red fluorescent protein. We can see that through the male abdominal cuticle. So I'm sorry, what you're looking at is the tail end of a fly. So this is outlining the abdomen. These are the little legs here. And so the head and the wings would be like off the picture here. So just to orient you, the abdomen of a fly here. Then we see that the RFP goes away when we do the temperature induction of damage. And then when we allow recovery for 5 days, we start to see that the RFP begins to recover. So we think that this will be a tool for us to be able to monitor the damage and recovery in the same fly, and even better, to perform a genetic screen where we could cross these, for example, to RNAi lines or look at different mutant backgrounds and see what might be increasing or decreasing either the rate or the capacity for regeneration after damage. So that's where we're going with this right now. Okay, so in my last couple of minutes, I just wanted to argue that there's a cell cycle transition that we should be aware of, and it's one that we probably haven't thought about much, which is this G0 to polyploid transition. So how is this transition out of G0 working? The cell isn't really re-entering this mitotic cell cycle. It's sort of doing this jump over to these so-called cell cycle variants, like the endocycle or endomitosis. Now in endomitosis, you get nuclear mitosis, but no cytokinesis. And so this leads to the binucleate or polynucleate situation. But in an endocycle, which is mostly what I've shown you today, you have a single nucleus. In the case of the binucleate accessory gland cells, what we see when we drive additional endocycling is that both nuclei within that cell endocycle become even higher ploidy. And so this is a truncated cell cycle where the cells go through a G phase, a gap phase, and then go through S, but there's no mitosis, no cytokinesis. And so they just increase their nuclear size, they increase their cell size, and they increase their DNA content. So why would the cell do this? Like, why wouldn't the cells, like, why wouldn't the tissue just choose to go back into mitosis? Why would they choose to go into the endocycle? Well, so in the two cases I talked to you about today, so one in the brain, you can imagine that mitotic situation for a neuron or glia with connections might be catastrophic. And so if you're losing cells, you may need a cell to be able to make more stuff and make more connections as a way to compensate for cell loss or cell damage over time. And so you could imagine that if a neuron has all these connections and it tries to undergo mitosis, that might be catastrophic in the brain. Similarly, for epithelia that perform barrier functions, we know that mitotic reentry for some of those cells that are forming a barrier can be catastrophic because mitoses breach the barrier, the epithelial barrier. And so just a reminder that part of what's happening in the male accessory gland is that that lumen is filled with sperm competition proteins. It's filled with antimicrobial things. It's filled with proteases and all sorts of stuff that the male is going to transfer the female that he may not want circulating in his hemolymph. So it might be really important to keep that barrier intact. And so that's one pressure for these tissues to use a postmitotic regeneration mechanism. But the other thing that I think about this is just from the cell cycle point of view, it's actually easier to transition out of G0 into the endocycle than it is to transition into a mitotic cycle. And the reasons for that are two. Again, inspired by Brian Calvi, he had shown through some really nice work that endocycling cells have oscillations of the cell cycle oscillator, so-called E2F oscillator, where it's turning on and off cell cycle genes. Mitotic cells have this oscillation too, but what he had shown was that cells that undergo the endocycle, they have what he called dampened oscillations. So basically the magnitude of those oscillations to drive the endocycle is much, much less than the magnitude you need to drive a mitotic cycle. And so you only need just a little bit of weaker E2F activity to drive you into the endocycle than the big rises and falls that you need to drive you into a mitotic cycle. So we think that the dampened oscillations is part of the reason why it's easier to enter the endocycle. The second part, this is work from Paul Nurse's lab in yeast, but still cool. And what he showed really convincingly, and we now know is the case in mammalian cells as well, is that the the CDK activity threshold, so just taking all the CDK activity together, the reason why S phase precedes M phase is precisely because the threshold of CDK activity that you need to trigger S phase is much lower than that you need to trigger M phase. And so if you're a cell that's in G0 and you're weekly reactivating your cell cycle, if you can only squeak out a little bit of E2F activity and a little bit of cyclic CDK activity, because you still have all these other differentiation things blocking your cell cycle, maybe you enter the endocycle instead of a mitotic cycle. So I have this philosophy that it's easier to transition from G0 into an endocycle than into a mitotic cycle. And so I have a fantasy that we are going to see, as people look for more and more examples of this kind of transition in mammalian cells, we're going to end up finding it all over the place. We're going to find it in kidneys. We're going to find it in all sorts of other tissues where the epithelia are performing barrier functions, but also where, they just can't reactivate the cell cycle to the degree needed to enter a mitotic cycle. So yeah, I'm just going to throw that philosophy out there, maybe much more common than currently appreciated. Okay, because we think it may be much more common than currently appreciated, a pandemic project undertaken by Shama Nandkumar and Emily Rose, which in my lab, both of whom who graduated, was to generate what we call the polyploidy atlas. So they were just combing the literature for examples of funky polyploid cells, like for fun. And we ended up putting this together into a big database that basically the back end is just a fancy Excel sheet, but we ended up putting it on the web. And so this is now in our lab website. You can go to the polyploidy atlas at polyploidy.dev, and you can search for references in the literature for different cell types, tissue types that become polyploid. We even have it separated out. Is the polyploid physiological or is it induced, for example, by damage or by oncogenes or things like that? So, and then you can also tell us if you want us to add your paper, so please let us know. And the idea is that this will continue to grow with time and that hopefully our prediction would come true that this will be far more appreciated than we realize. Okay, so with that, thank you so much. These are all the people who actually did the work. And I highlighted work for you from Shama, Jamie, and Allison, Chelsea, and Emily. Our work on the accessory gland, we're about to update it, so you might wait a couple weeks, but this is a QR code. This is our manuscript number if you wanted to check out the data on bioarchive. And I also have to say the Drosophila community is super awesome, as some of you fly people know. And so I want to point out Don Fox's lab kind of shared with us the demise flies, without which we couldn't have done any of the accessory gland regeneration stuff. And Ishwar Hariharan's lab developed the coin flip flies, which were essential for us to identify the polyploid cells in the brain. And so I'll take some questions if we have time. Yeah.
Very nice seminar. So I just need some clarification. So I really like the model that you have threshold models and how they can re-enter the cell cycle. But going back to the idea of this being something you see in aging organisms stuff. You alluded to the idea that becoming polyploid may make these cells, certainly in the brain, more functional. Did you, I mean, you tried to say it's something that maybe needs to happen, protective, it happens because, and I was wondering if you could expand on that and to flip that, if you could, or if you're starting to imagine ways to prevent it from happening and see if you get flies that can't live in song.
Yeah.
Or have, I don't know how you measure fly brain activity, but it's stupid faster.
I don't either, but fortunately I have neuroscience colleagues. These are great questions. So this is exactly what Dina, the current postdoc, is working on. So we know, so we tried a myriad of ways to try to increase or decrease the level of polyploidy in the brain. Long story short, what worked is that if we knock down CDC6, which is one of the replication licensing factors, we can reduce the polyploidy. If we overexpress geminin, the replication licensing inhibitor, we can increase the amount of polyploidy. And so she's hoping to use those genetic manipulation tools to try to actually look at, for example, a subset of neurons or either increase or decrease the
So it's very
ploidy.
interesting. The ones that you manipulate are both directly involved in controlling license.
I know. And we tried many orcs. We tried quite a few things to manipulate the polyploidy, and it was CDC6 and geminin that worked.
That's really interesting.
Yeah, we wonder if there might be some sort of very strange replication complex that's like not the normal replication complex in this scenario. And I should say, we also don't know that the full genome is being replicated.
Yeah, no.
Yeah, so, but yeah, we tried manipulating many different components that we thought would alter the ploidy, and it was really only CDC6 and geminin where we got reproducible and statistically significant effects.
Did you ever try CBT1?
We did. I don't remember off the top, but we thought that one would work as well. Yeah, and I don't.
No, this is not published.
The CDC6 and geminin data is published. We use that kind of as a tool to look at the degree of polyploidy in the brain.
And so then back to that functional idea. So
Yeah.
what's your idea of you said something about them making more stuff. But
Yeah.
that's just not very satisfying. I don't mean to be rude, but
Yeah, so, well, when you look at the fly brain, there's these insulin producing neurons in the central brain, and they're very large and they secrete a lot of stuff like these Drosophila insulin-like proteins. And it's been kind of speculated that they might be polyploid because just kind of sheerly because of their size. And so we were thinking maybe like you need to secrete vesicles, communicate, the neurons need to increase activity in response. Because how does the brain still function if like 5% of your cells every week are being lost due to damage and aging? We do see, dead and dying cells when we do these assays in the aging brain. And we also see from our flow cytometry counts, we know that the cell number decreases in the fly brain with age. So those cells are having to, I guess, like
the
pick up some
fractions of the
slack.
cells become polyploid and get bigger.
Yeah. So maybe they're compensating for the loss of activity by the other cells. The other thing this was inspired by,
It's just when you think about, polyploid microorganisms, I'm talking all this stuff. In microorganisms, you think about problems too, right? I mean, you can tolerate it to a large degree, but it's not problematic.
and that's actually kind of what we want. We wonder if maybe initially it's actually helpful, but then later as the fly ages, it might be damaging, because you'll notice our polyploidy, it always plateaus at the later so-called geriatric stages for a fly. And we don't know why that happens. Like, why doesn't it continue to increase? It plateaus, and we're wondering if some of those cells are starting to die with age, like the polyploidy becomes a Achilles heel or something.
Fascinating, thank you.
But total speculation. I have no data to back.
Yeah, no, that's what.
Yeah.
Yeah, I have a question about sleep. So the double strand breaks are associated with wakefulness in neurons, and I think the hypothesis that sleep evolved so that it would enable single neurons to perform nuclear maintenance, specifically like repairing double strand breaks.
Very
So
cool.
you looked at whether if you keep the flies from sleeping, which decreases their lifespan, you get that would increase the number of
We haven't looked at sleep at all. We haven't looked at sleep at all. And also, I mean, as an extension to that, I know that there's this now newer, well, so R loops in active neurons is something that could also be leading to DNA damage. And then in addition to that, activity related DNA damage is a thing in the neuronal field, right? So, no, we haven't looked at sleep. We could. We have a collaborator at U of M who has some experience looking at that, and we should do it. We need, yeah, more people. That would be really cool.
I just have a really quick question. You mentioned that it's much more common in the optic
Yeah.
lobe than that is in the kind of having an eye, but why do you think that would be more common in that part?
Yeah. I mean, it's all total hand waving, but just thinking maybe that's where most of the activity is. I don't know. I've never done like G-camp imaging in the fly brain, but it seems like they need their visual system to constantly be active.
But it doesn't require
Yeah, I guess I don't have, I got nothing for that. Yeah. No, that's a good point.
Sorry, waiting, so.
there must be something different than the
So I remember the other thing I want to say, there is a paper from Eduardo Moreno's lab where they actually saw a population of what they think are neuroblast stem cells in the Drosophila and then in the Drosophila adult brain in the optic lobe. We don't know if that could be there. I should say the number of those stem cells that they found in their paper would be so small compared to the number of polyploid cells that we're getting that, I mean, maybe they come from those, but it would have to be like through several divisions. Yes,
Danielle asked my question.
Oh.
I can follow up and ask whether you've ever put the flies in the dark.
yes, we did do that one. So we put the flies in complete dark and the polyploidy was the same. The one we haven't done is complete light. And we've been talking about doing it for a while, but because it was easier to find an incubator that was dark than one that was completely lit, we did that one first. But yeah, I mean, hyperactivation and whether that could be correlated is something that we should look at. Yeah.
Do you have any other information in driven neurons of like what types of neurons might be more specifically might be that you think that this is biased with particular subtypes of neurons?
I do. So this is super preliminary again for the recording, but so we've started to look at different drivers that drive in neuronal subtypes in the optic lobe. So like looking at like the L1, the TM, like the different classes of those transmedulla neurons, the different classes of the lobula neurons. And so we are seeing some differences depending on which class we look at. And it does seem like the bigger cells are more likely to have more polyploidy, like the ones where they have to, the connections that they're making are like fully across the medulla are more likely.
We have one question online. Christopher McAllister asks, thanks for an interesting talk. Have you coordinated with labs looking into other organisms to assess if there are similar neuronal and accessory gland polyploid genotypes? I.e. in nematodes or planaria, how does salivary gland polyploidy compare to that in neuronal and accessory glands?
Yeah, so the salivary polyploidy is way more. So like we're talking about ploidies in the 4C, 8C range. For the accessory gland, we're talking about like 6C to 12C range. So much less than the salivary gland. Other organisms, so actually there is some research in mammals about polyploid neurons. And so there's some recent work showing that there's a subset of neurons in the rat brain that are tetraploid. And then there's also some work from Alice Davies' lab in France on polyploid neurons in the mouse brain. We've been looking at other Drosophila species, and again, super preliminary, but we see that different Drosophila species have different levels of polyploidy, and some have very little. So it's not, yeah, so there seems to be some interesting effects there. And then we also looked at other Drosophila species at the accessory gland. And we suspect that those also are poised to endo-replicate. We see endo-replication in other Drosophila species accessory glands as well, but we just don't have the genetic tools in the non-melanogaster flies to be able to induce the damage. Yeah. I haven't looked at planaria. That would be fun.
Let's maybe last question and then.
I was wondering with the, accessory gland damages, have you done them at different age?
Yeah.
And then did you see any differences in coloring?
Yeah, that's a great question. We desperately want to do that because our hypothesis is that the animals lose their capacity to recover with age, right? I mean, that's the ground state hypothesis. So we ran into a problem with the demise system, which is that it is somewhat leaky with age. And so in the early stages that I showed you where the male is young and he's, you know, gaining his fertility, we can do this very well. But in the older flies, we've been struggling with technical problems. So we're hoping to like find the right driver damage combo to make it not leaky in the aged flies.
All right, let's all thanks Laura again for